Our next session to kick off the afternoon is HD Insights of the Year with our CEO, Dr. Daniel Clausen and Dr. Dietrich Habenberger from Neurotherapeutics. HD Insights is a semi-annual publication of the Huntington Study Group. You also should have received a copy in your swag bag when you checked in, so you should have a copy in there. All right, please welcome Dr. Daniel Clausen and Dr. Habenberger to present HD Insights of the Year.
Great to see everyone again. Sorry that you have to see my face again. Just a couple updates on HD Insights. We just put out a new edition. I think it's being printed now, but it really talks about some of the early changes in HD and highlights some of the neuropsychology working group work that's come out of this effort to try and define non-motor presentations, also psychiatric presentations. And we have some stories from patients and interviews with physicians. So it's a really pertinent for this morning's session. So HD Insights, if you haven't been to it before, it's usually a little different than you typically get. It's not necessarily, about mutant Huntington or wild-type Huntington or NFL, all those things. We've had different speakers like Bruce Compass, who's done a talk on stress. We've had Angelman syndrome talk. I gave a talk last year. And this year, I thought it'd be really great to have a personal story from Dietrich Habenberger, who is the executive medical director at Neurotherapeutics. I first met Dietrich, I think it was on a phone call, when he had just joined Neurotherapeutics and we were getting ready to start the Ingrezza study, Connect HD. And he sounded like Arnold Schwarzenegger, because he's from Austria. So every time I think about, he lives in San Diego, and him and Arnold must hang out together, right? But Dietrich's a really interesting guy. He's a singer. He sings in different choirs, and he's sung with Dudamel before, which is really, he's got a lot of artistic talents, like most Austrians. And he has had a really interesting journey in terms of his academic, government, and industry journey. And I think he has a lot of really interesting insights about therapeutics. I mentioned earlier the desire to find fast, efficient trials, and he has thought a lot about this. And I really am looking forward to hearing his insights on this topic. Before I bring him up here, I just want to make one other announcement, and that is we're fortunate to have a new editor of HD Insights, Leroy Fox, who is maybe here, and Margie will stay with. I'm going to do other things with that. But aside from that, Dietrich, we're so grateful that you're here, and look forward to hearing your insights.
Thank you. Thanks, Daniel. It's an absolute pleasure and also honor to be standing here and to speak to you for the next 20 or so minutes. Oh, clock's down there. And also, excuse me that I'm going to take off my Neurotherapeutics hat for a moment, and as Daniel asked me to, essentially share what I think, what moves me currently in my job. And my job, I don't define that necessarily as my job as role as executive medical director at Neurotherapeutics. Yes, that's my job, but I think we all have a greater job to do, right? And our job collectively, I think, is bringing better treatments to our patients who are in unmet need, and do that way faster and way more efficient. So that's something that gets me up every morning since many years, probably 2 decades now, and just want to share a few thoughts with you on that. I've been asked to put the slide in. I don't know if we can have a Q&A after that. I'll be happy to. So it is titled "My Hopes for Neurotherapeutics," and I'm actually also super excited to do that here at the Huntington Study Group meeting because the Huntington Study Group specifically, really had been a very important part of my life, essentially, since the past 5 years. Daniel mentioned we had a call shortly after I actually joined the company, and actually this was my first industry job at all. So he was one of the first people outside of the company that I interacted with, and we were immediately in the zone. We were completely clear what we need to do here, right? And this is something that goes back many years also from my end, because I've been, in my past, I've been the EHD Insights person in Medical University of Vienna in Austria. I've been to EHDN meetings very early on in my career. I've been taking care of patients with HD for many, many years. Actually, one of the reasons why I joined Neurocrine is, somebody of the name of Chris O'Brien, came up to me at a poster at Movement Disorders, I think in Buenos Aires or something like that. I was presenting a poster on an electrophysiological biomarker in hyperkinetic movement disorder, and that was about tremor. He was really interested in that. He dug in, and he had the right questions, the methodological questions. Said, yeah, this is a nice academic interaction. Then I realized, wait a second, he's an industry guy, but his background, of course, is in academia, is in the Huntington Study Group, and eventually when I then saw how Neurocrine was doing, as I was looking at many other companies, I was really excited to see that essentially it's the same crop of people that work on the same job that we all have together. So that's essentially where I am right now. And so what I want to share you a little bit what I'm thinking right now about neurotherapeutics, and then essentially going forward, what are my hopes. So as I mentioned, this is not a Neurocrine presentation. Full disclaimer here. I'm not going to talk a lot about, or at all about, our trial or the results. There were excellent presentations earlier today. There are some posters out there. And of course, as a disclosure, I'm a full-time employee at Neurocrine, and I also do other stuff. I'm a voluntary associate clinical professor at UCSD, where I'm happy to help out the junior faculty in neurophysiology, for example. So lots of cool stuff there. Going back, yes, I'm from Austria. I hope that I got rid of my accent a little bit, but I'm still a little bit proud of my accent. Having been actually in Sacramento 5 years ago, having been my first HSG meeting was fun, because that's of course used to be the place where the governor was in office. It was also very fitting to have our first investigator meeting at 2019 in Sacramento at the movie theater at the IMAX there in Sacramento. Victor and I were sharing some photos last night, and to be able to stand there at the investigator meeting and say, "I'll be back," was just fun. Okay, let's leave that. let's leave that aside. So I did my training in Austria, went to medical school, started my academic path on a tenure track, or actually did first my residency, focused on movement disorders really early on. And as you usually do, if you're really interested in movement disorders, you pursue that track. You go on to do a fellowship, which I did at the NIH. So this was my first move over to the US in 2008, started a postdoc fellowship at Mark Hallett's lab at NINDS, focusing on neurophysiology. Focus was also on tremor in this regard. I've worked on that topic before. Then 2011, after 3 years, went back to Vienna. So my position was still there, went on a tenure track, became associate professor on July 1st, 2014, and on July 14th, I left again to take a job offer to come back to the NIH. And the backstory of that is that my fellowship project essentially was a clinical trial. This was not my choice necessarily, but this was the project that was available to me. I was working in essential tremors, working in phenotype, genotype aspects. Claudia Testa is here. We worked together on the first genome-wide association study in essential tremor. I was back in Vienna. So I was really interested in the genetics and how genetics relate to phenotype in patients with movement disorder. And Mark Hallett essentially said, we have this experimental compound, it's a long-chain alcohol, We want to test it in essential tremor. So, do you want to, test that? I said, yeah, we would be interested in doing that. Would be a nice thing to learn. One step led to the other. It was a phase 2A type of, now we would call it phase 2A, 1B type study. Single dose, very PK heavy study. My first clinical trials methodology course, Carl Kieberts was my mentor. Another connection right there. And this essentially opened my eyes to say, okay, that's it. This is my methodology. This is my technique that I really want to pursue going forward. So I was basically, I decided at that point I'm going to be a trialist. So 2014, NINDS essentially reached back out and said, hey, we want to build up a clinical trials unit here at the intramural program at NINDS. Would you be interested in running that? We don't have that many people who are actually interested in running trials, thinking about trial design, outcome measures, etc. And you seem to like that, so would you want to do that? And essentially said, okay, I'm going to move over. So I essentially gave up my tenured role there in Vienna after only 2 weeks, moved back to the US. And then 5 years later, not necessarily was looking for other jobs, but one thing that was important to me to see and also to observe, one element is that at NINDS we built up a fellowship for clinical trials methodology and regulatory science together with the FDA. So fellows were able to, still are, it's an ongoing fellowship, can join NINDS, NIH, rotate through intramural and extramural, and also spend a year at the FDA. And I was fellowship director at that point, and we were test running this fellowship, so I spent a few months over at the FDA, and then I realized, looking at the applications that coming in from industry and from other places, where actually treatment development is happening. So then I really realized that the last piece of the NIH mission to bring basic discovery to our patients, that last piece to bring it to our patients, this is really where industry is really good at, and that's what they do really well. So eventually, I made that move to Neurocrine in 2019, and this is where I am. And these are nice pictures from Vienna, the NIH, and now in San Diego. So this is kind of the academic background along the way. We published lots of stuff. You see at the beginning we worked on GWAS studies in essential tremor that led to clinical trial work and also a deep interest on my end in the context of the discovery, development, and validation of biomarkers that help understand whether we are hitting our target, whether we're engaging our mechanism to come to good, early, and smart decisions in our clinical trials. And that's probably the only Neurocrine content that I have here. It's that our really beautiful Lancet Neurology paper on our CONNECT-3 trial, really led by Erin Verstimming and her team, including Daniel, which demonstrated the efficacy and safety of valbenazine in the treatment of chorea with HD. Let me zoom out. Big picture. And, I'm a neurologist by training, general neurologist, and focused then on movement disorders. But, you hear always these news, company X pulling out of neuroscience, company Y going in, etc. Lots of debate around where treatment development is going, but the fact is that neurological diseases are a challenge for society. Most clearly, we're talking about an aging society. We're talking about, especially when we think about effects of neurodegenerative diseases on our society and also cost on society is undeniable, right? And I already used the word cost here. Don't try not to be too businessy here, but I think Claudia Testa brought up a really good point before. So who's going to pay for that? Who's going to pay for our care professionals? Where are the resources coming from? So it's very clear, and we see that in HD, and we see that across the board, is that there's lots more resources that are necessary to care for and treat our patients with neurological diseases. And how is neurology doing so far? How is neurotherapeutics doing so far? Yeah, it could be better, right? If you look at just the metrics, you start with the metrics, is that, of course, neurology, not necessarily of course, but nobody would be surprised, is that neurological treatments are not the ones that are on the top of the most likely to be approved treatments by the FDA. It's led by oncology, infectious diseases. Only 12% of FDA approvals, at least in that time window, 15 to 20 are in neurological indications. And when you think about treatments that go into human testing, into phase 1, less than 10%, and actually I have to graph up here, it's a little small probably on the screen here, is only 5.9% of compounds in neurology that enter phase 1 actually reach approval. So you have to think about the attrition that's going on there all the way, and so it is it's much more likely that a treatment that gets into the clinic fails just by simple math here, then it's going to be successful, right? And then it's taking a long time. The median duration is about 11 years currently to get from phase 1 to approval. So when we're excited about new treatments entering the clinic or about to enter clinic, we have to think about when will that be available to our patients, right? Our patients need treatments now, right? Better treatments right now, but we all have to work fast and hard that we're beating these 11 years, right? Because I don't want the exciting treatments that we talked about earlier today not being available to our patients until sometime in 2035, right? So. And there's some differences, small molecules actually have a lower likelihood for success than more advanced and more targeted treatments like gene therapies, for example. So there's some hope that with targeted treatments we can increase our likelihood for success, and we hopefully potentially also increase the time to approval. So what we need clearly is an increased efficiency in our treatment development paths for neurological diseases. Digging a little bit further into the numbers, your chance of success increases the further you get along your treatment development path. So if you are in phase 2 with your compound, you have a 12.3% chance of likelihood to get approval in neurology. 46% of compounds that get into that are in phase 3 to approval. Think about that. Less than 50% of compounds that actually enter phase 3 actually reach approval in neurology. We should be able to do better here. That's my view. All right, let's talk about money. That's certainly the elephant in the room here, right? And I really not only congratulate Daniel to his new role here at the HSG, but also he put his finger right on the pulse to say we need to do our trials more cost efficient, and we need to do them faster. And again, there's a triangle here always, and you can always and the triangle is cost, time, and the other is quality, right? So we cannot compromise on quality here as well. So you have to think about to keep that all into context. But currently, again, this is benchmarked, it's out there, it's published. The median cost of a drug to be developed in CNS disorder is about $750 million, right? Can you imagine that? And that's the median costs. Gene therapies, for example, are more on the more expensive side. So for somebody who embarks on treatment development, you have to know what you're getting yourselves into. And if you think you want to reduce that, and if you think you want to make it faster, then let's think hard to come up with a plan. How are we going to do that to be successful and not to compromise on quality, right? So why is it that expensive? Of course, there are high attrition rates. If only every other compound that gets into phase 3 gets approval, that cost has to be rolled in. For example, we have lengthy trials, and there's extensive research that is necessary. And I'm gonna basically put 4 ideas out there that I'm gonna walk through a little bit in my next few slides. So how can we improve? What could be potential ways to do that? Paths forward that could inspire us to think harder to figure it out specifically for HD in that example. So we want to think about innovative trial designs and innovative development pathways. We need to improve our cost effectiveness of drug development. We need to build partnership and maintain partnerships across all the stakeholders in that process. That's academia, industry, regulators, and advocacy. our patient community. And then also, one thing that's really near and dear to my heart is we need to invest in our workforce of neurotherapeuticians. It's not something that you can just do on a Friday afternoon when you just get out of clinic. Starting off with development pathways. There is the traditional development pathway that essentially I've mapped these percentages against. Before you start with your phase 1, and then you go into your phase 2, then you go into your phase 3. You typically 2 well-controlled and adequate studies. Then you submit for your approval. That's a classic development pathway. Phase 1 typically conducted in healthy volunteers. Phase 2, you get into, you understand more about the activity in your patient population, target population. You understand more about dosing. You understand more about the mechanism of effect until you really get into phase 3. Phase 3, by definition, is the confirmatory stage of drug development. Recently, FDA has been really being on the forefront and pushing sponsors and pushing the field to think more about accelerated approval pathways. What does that mean? Accelerated approval, you receive accelerated approval based on a biomarker, for example, that has a reasonable likelihood to predict clinical benefit. And that's actually not just something that I came up with, the reasonable likelihood. There's actually a definition of what is a reasonable likelihood to predict clinical benefit. So that could be a biomarker-based approval, for example. So you need to link basically your biomarker signal to a clinical benefit that is to be expected in your target patient population based on your therapeutic mechanism of action. And I'm going to talk about that a little bit in a few slides down the road. For example, also in more advanced therapies, gene therapy programs, for example, you don't do your phase 1 study in healthy volunteers. This is something where, this is typically done in small molecules where you start in healthy volunteers. Gene therapy programs typically, as you know, as we have discussed here at length, start collecting safety and pharmacodynamic and target engagement measures early on in patient populations. This gives us the opportunity already in the target population to collect biomarker data that could be potentially used to support accelerated approval. So there are programs out there that could reach a stage of an accelerated approval already after your phase 1/2 type study. And then, it is an accelerated approval. It's not a full approval. So typically, it is required for programs like that to still continue in phase 3, like clinical trials or programs, to then essentially ask for conversion into a full approval. So it doesn't absolve you from the need to actually to do your phase 3 trial. But you already received approval. So that can potentially increase and accelerate your timeline to get to approval where your compound can be prescribed to patients. One example already has been mentioned earlier before, the tofersen example in ALS SOD1 population. So there are 2 examples that I want to bring, one of them being the ALS example. It's been already nicely presented by Christine Sampaio. I think one additional point that I want to add to that, so this is a program that was approved, received accelerated approval. I think that's an important distinction here. Received accelerated approval based on NFL, the biomarker. And the clinical evidence not only came from cross-sectional data that was available at that time, but the clinical evidence also came from the program itself, right? Because the program itself demonstrated in their open-label program that patients treated with tofersen in the open-label program essentially ended up with a better clinical outcome than patients who were treated on placebo first. So I think that's an important one. So there's in addition a prospective evidence for NFL as a biomarker that has a reasonable likelihood to downstream will confirm to clinical benefit. But again, this is an accelerated approval, not a full approval. FDA retains basically their right to revoke accelerated approvals, for example, if that reasonable likelihood doesn't turn out into actual clinical evidence. So that's something that we have to be aware of. On the other side, there are programs that receive full approval, not accelerated, but actually full approval. One of them, interestingly, not necessarily based on a biomarker, but based on a single placebo-controlled trial with an additional open-label extension is omaveloxolone, which is a treatment that has been approved recently for Friedreich's ataxia. And the primary endpoint was the modified Friedreich's ataxia rating scale, the mFARS. And what weighed really heavily in the FDA review document, and I actually can recommend everybody to actually go in and read these publicly available FDA review documents. They are really interesting documents. Is that this basically the evidence came from the randomized controlled trial, supported by basically by a natural history study, long-term data compared to a natural history study that the community was maintaining over many years, the FR-COMS trial, a natural history study that was really rigorously conducted to essentially serve as a natural history control arm. Often we use the term natural history study not very cleanly, and it's often kind of a mixed bag, and some people don't even like the term natural history study. That in that case, it was really we can view that study as a prospective, rigorous study to collect prospective data that really reaches regulatory grade data quality to serve as an external comparator for, in that case, full approval. So that tells you how important biomarker work is, that to provide the data package and the evidence that can convince regulators that your biomarker has a reasonable likelihood to predict clinical benefit on the one side, and on the other side, to conduct natural history studies in a rigorous way that could support approvals of compounds. So yeah, these are examples for more novel pathways here. How can we improve cost effectiveness? Of course, if it were so easy, right? We need to improve our target validation, the targets that we're following. We need to have really good confidence about before we move forward, or essentially have to be aware of the risks to move targets forward into clinical trials. Typically, this is where it gets expensive, where we haven't had very clear validation about the target. So that's certainly, often, we take these risks and move forward, but then, the field figures it out as we go. But that's certainly something that we have to be aware of. What is our conviction and what is our evidence before actually we go into clinical trials? What the field likes to do is conduct early proof of concept studies, if possible. I brought an example here. Eli Lilly put a whole unit in place there. The charge of that unit was to conduct essentially trials or studies or clinical programs all the way to proof of concept. It's essentially get to proof of concepts early and fast, and then make a decision before you get into later stages. Typically, your phase 1 or early phase 2s, this is your learning phase. This is where you can also map your result that you get out of your trial against what you initially set out to do. You can define your effect size that you say you define as your critical success factor to move forward into a downstream phase, and you essentially make decisions right there. And that increased their success rate from phase 2 going forward. So that's a model. We already talked about optimized trial design. That also includes seamless phase 2 and phase 3 trials. The risk here, of course, is that it also then takes greater upfront investment. So it can be balancing against the other risk in terms of the clinical development cycle times. If you go by your traditional model, phase 2 followed by a phase 3. What industry does not like is what we call white space. That's the time in development programs that's between clinical trials, where essentially you don't enroll the patient, where you don't move forward. That's typically the time that's necessary to engage with your regulators, because the FDA wants to talk to you in a end of phase 2 meeting, for example, to agree with you on an endpoint for a phase 3 trial. That's typically something that you can only do when you actually have data in your hand from your phase 2 study. So that's just something where it's a conundrum, right? It adds to time, it adds to therefore also cost, and it moves down your potential approval timeline. So that's what I mean by improving development cycle time. shifting attrition to earlier stages, making hard decisions, right? If you're not convinced, that you can move into what we call a confirmatory phase, right? Then what do you want to confirm, right? To make decisions early on. Networked R&D model is basically how, who else has skin in the game? Who else can you partner with? Who else can, don't just say share the cost, but also share, your effort to be partnering here. And that's actually a really good kind of segue here to that element about partnerships in treatment development. And there's no better place to talk about it than here at the HSG annual meeting, because this is really a very good example where collaboration between a sponsor and the HSG is a good example for a joint partnership. Because we all have the same interest in place, and that's a difference, right? That's a difference between working with HSG than working with a quote-unquote commercial CRO, right? Because that a commercial CRO that does not have quote-unquote skin in the game. So this is really important, and there are multiple organizations here in the HD space, CHDI, HDSA, and others, of course, but that also extends to other opportunities, pre-competitive consortia, CPATH, et al. Public-private partnerships between government and academia, license partnerships, et cetera. And of course, the activities around patient-focused drug development with the patient community to partner here early and often. There are examples for that also in how successful that can be, in my view, and how that can actually lead to significant impact in the field. Epilepsy is one example here, for example, where the community actually got together 5-6 years ago and was thinking about, was worried about rates of SUDEP in placebo arms in epilepsy clinical trials, where industry got together with academia and regulators to say, is it actually ethical to do a placebo control in epilepsy, for example? And essentially, it was decided to put something together. It's called the Epilepsy Research Roundtable, where a topic that the field really concerns, and the field meaning academia, industry, government, and patient advocacy, and put that front and center. You get together, you get your data together, you do that behind closed doors, and you essentially try to listen to each other and try to come up with a solution. Here in that case, this is an example of a recent one, where there was the question, can we extrapolate the efficacy of epilepsy clinical trials, phase 3 clinical trials that are typically done typically done in adults or adolescents? Can we extrapolate that efficacy data down to the very young patient population? Seizures, something that happens often right after birth. So if can the efficacy of anti-seizure medication, can that be extrapolated down to the age of 1 month of patients, of children with epilepsy? Instead of having to do basically an efficacy trial in your patient population, it was often very, very difficult to do. And that was debated, discussed, data was presented back and forth with regulators, with academia, with industry people in the room. And that eventually led to a publication, but also led to an FDA guidance. So essentially, this is really something that the industry now is bringing, can bring forward. If everybody agrees, this is how we do it. This is how we do it going forward. And having experts in the room to align on that, I think is really powerful. And that also reminds myself then, it's essentially the same kind of people, right? That work in academia, government, in industry, work together essentially to improve how we bring better treatments to our patients. Brings me to my last segment here, which is near and dear to my heart, is building a neurotherapeutics workforce, right? Because at the end of the day, we're motivated by the same thing. At least that's what I think of myself. That's why I went into clinical development and work in translational biomarkers. Whether I'm doing that in academia, industry, or government is one thing. So I do truly think that there is a room for something that could be, something like a subspecialty of experimental neurotherapeutics. We need people that come out of, of neurology training, have neurology background, child neurologists, adult neurologists, neurosurgeons, advanced practitioners, et cetera, that really double down and understand our patient population, understand the need, but also have the appropriate training on how to do it, on the methodology, right? That includes biostats, that includes regulatory elements, et cetera. So really, my vision here is really that you can select that as your subspecialty, and you go into that, you do your fellowship, and then basically you are one of us, right? Going in academia, in industry, or in government, and so we are all on the same page and can work together ultimately on the goal to bring better treatments to our patients. There are examples out there already mentioned. The NINDS FDA Clinical Trials Methodology and Regulatory Science Fellowship that I can really recommend. There are others in academia, University of Rochester, MGH, really on the forefront. I also think back, Ira Scholzen was certainly somebody who was really very active and very, a big driver of careers and career development in neurotherapeutics, and I certainly was able to benefit from that also a little bit. There's lots of literature to get out there. I would really recommend you dig into that if you think that's something for either you yourself or for your mentee or for somebody who's coming up in your ranks, somebody on your team who wants to spend a little bit more time on that. The field needs you. There are grants out there. There are funding opportunities out there. There are good organizations out there, for example, like here, and make a little bit of a plug of a meeting that's coming up. It's the American Society for Experimental Neurotherapeutics. It's happening every year in Bethesda. It is essentially, a professional society of neurotherapeuticians out of academia, industry, and government. And so if you're in the area, stop by. There's also for junior folks a really good clinical trials course that I can recommend you guys participating. And with that, I'm actually a little bit over my time, apparently, but with that, I say thank you. Thank you for bringing me out to Cincinnati. The picture here was taken right this morning at sunrise when I took a nice run here around town. It's really an amazing place to be. It's an amazing community to be in, and I hope you guys knock on my door at one point if you come to San Diego, which is the picture on the right. Thank you.
Since you called out my earlier question, I'll just ask a different version of it. You cited several truly important things that I have found it extremely difficult to get funded. One of them is high-quality longitudinal observational databases. We do have Enroll now, but that wasn't true for decades, and is not true for many other disorders, right? But getting that FDA review was very interesting. They cited something that people had really had to kill themselves to fund over the years as key data for getting that approved. So funding longitudinal studies, finding the clinical trial participants, particularly in HD, it has been striking over the last 2 days, the breadth of opportunities coming down the immediate pipeline, which is incredibly exciting, but also really striking how they're all looking for the same tiny little group of people. And doing outreach, doing case management support, finding those people, getting them plugged into clinical care, identifying new diagnoses in families, and getting them to a point where they have the bandwidth to interact with clinical trials, all of that is work that I don't have funded. And finally, yes, I would love that kind of fellowship at many more places than the ones you listed, but you did list a fairly tenuous and somewhat non-reproducible set of funds for those fellowships. So possibly putting your neurocrine hat back on, what would you speculatively say could be the role of industry in helping fund efforts that are not possible to fund off of clinical care earnings, but are literally critical to our success in the next 5 years of accomplishing these clinical trial experiments that will get us the therapeutics we want.
Yeah, absolutely. Thanks, Goya. So, to answer your question, I'm gonna take the hat that you put on my head right off again. I'm gonna put it there. No, absolutely. And I probably preface it with one thing. One thing that I really appreciate working in industry, and that's probably one of the eventually the things that at the end of the day makes me really happy, is because there's one thing, there's one goal in industry, or at least in the job that I have. We prioritize everything to get to an answer, to move that thing that we have a belief in forward. There is, and that also sometimes, it's probably, the FDA package that we submit to get to a decision, to get to our next trial, has a higher priority than the paper, right? Sorry, that's our job, right? This is why we have a partnership, et cetera, because it's everything that gets us to the next point, to get to a decision. Is this going to work or not? And if it's going to work, how can I move forward faster? If you, and I think this is why I brought these examples, I think now industry is very well waking up and say, hey, wait a second, there's now a drug that's been approved based on a registry, based on a natural history study. So suddenly, you will see that industry will go out there. So, okay, do you guys have that? Can we do that? Can we do that? But can we do it like fast and tomorrow, right? So I think if you think about, if you try to put the Neurocrine hat on, is that to say, what would we like? Yes. So if you work in a condition that could potentially have a study or basically a cohort like that ready for me to submit for approval in 2 years or 2.5 years when I'm done with my trial, then we're talking, right? So that's one thing. And you also mentioned recruitment. Absolutely. I mean, people know here in this room. I mean, Daniel knows Addis Mendizabal is here. We talked about, where are our patients, right? For whom do we do our studies, right? How representative is my sample? And this is purely, it's in line with eventually that section that I have to fill out in my FDA approval package, and to say, how is your sample representative of the entire population, right? So I have to get data around that. That's number one. And number two is, if you tell me that there's a way to reach out to actually bring it to patients that otherwise we were not able to bring our trials to, then basically then I say, okay, how are we going to do it? Let's talk.
Sorry. Thank you. We do have to keep this going. Thank you so much. Thank you for your question. And our thanks again to Dr. Clausen and to Dr. Habenberger for HD Insights of the Year. Give them one more round of applause, would you? Thanks.